Hi, everyone. Thanks for joining today. We'll go ahead and get started in about 5 minutes. Hello, everyone. Thank you for joining today, and welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller. I am a NOAA Omics specialist, and I sit in NOAA Ocean Exploration. I didn't realize I was on mute. Omics, which is a suite of tools used to analyze DNA, RNA, and proteins and metabolites, has become a large priority here at NOAA in the past few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. This seminar series will be made available on our YouTube and our NOAA Omics website, and there is a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation. And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Biosensors of Environmental Change, Linking the Global Marine Microbiome with Ocean Processes by Dr. Alyse Larkin. I'm going to go ahead and share the presentation or share the screen with Alyse while I read a short bio. All righty. Alyse, if you want to go ahead and pull up your presentation. All righty. Alyse Larkin joined NOAA, the NOAA Global Ocean Monitoring and Observation Program in UCAR CPAS in December 2022 as a program manager for the Global Sea Level Observing System, or GLOS, and the Biological Global Ocean Ship-Based Hydrographic Investigations Program, or BioGoShip, in the Ocean Carbon and Biogeochemistry portfolios. She received her Bachelor's of Arts in Environmental Studies from Vassar College and her PhD in Marine Science and Conservation from Duke University. Additionally, Alyse completed postdoctoral training with Dr. Adam Martini at the University of California, Irvine. During her PhD and postdoc, Alyse worked with genomic big data to assess the interactions between marine microbiomes, ocean biogeochemistry, and environmental change. Thank you, Alyse, for presenting today. And I will pass it off to you.
Thank you so much for that kind welcome, Nicole. I just want to check, can you see my presentation?
Yes, we can see it.
Okay, great. All righty. Hi, everyone. As Nicole said, my name is Alyse Larkin. I'm a program manager with the Global Ocean Monitoring and Observing Program at NOAA, as well as the cooperative programs for the advancement of Earth system science at UCAR. I want to start out by thanking Nicole for inviting me to talk today. I'm really excited to speak to all of you about how we can go about linking the global marine microbiome with ocean processes. And to start, I really want to acknowledge current and past members of the Martini Lab at the University of California, Irvine. This work was done as part of my postdoctoral research there. And so there's a large number of grad students, other postdocs, project scientists, and undergraduates who contributed to this work. I also want to thank the US GoShip and Plymouth Marine Lab British Antarctic Survey science teams, captain and crews, and funding from the National Science Foundation and NOAA. So let's get started. Over the past 40 years, scientific research has increasingly recognized the importance of the global microbiome. And microbiomes are defined as the community of microorganisms found in a particular environment. Now, microbes have colonized pretty much every habitable surface of our planet, from the deep sea to the surface ocean to terrestrial and host-associated environments. And there's even some evidence that microbes can survive in the atmosphere. Moreover, over the past 20 years, the omics era in microbiology has resulted in really an explosion of complex and interrelated data sets. Now, an advantage of these data sets is that we can take a deep dive into them and ask a nearly endless array of questions about microbial biosensors across study systems. In particular, we can open that black box of microbial community function and diversity and assess its impact on environmental and host-associated processes. So these molecular techniques have revealed a huge diversity of microbes. Specifically, what we have learned is that the fast division rate of microbes leads to rapid adaptation and high diversity. For instance, we can take a look at this revised phylogenetic tree of life and see a huge number of branches that are in the bacterial clade. In contrast, these clades here represent all of archaea, and this little branch over here is all eukaryotes on the planet. So microbes, as I said, bacteria in particular, are highly diverse. And so given these fundamental properties of microbes, I'm interested in answering the question of how we can use this vast diversity of microbial genomic content as a biosensor of environmental change in ocean processes. So in my talk today, I'm first going to provide background on my main study organism, Prochlorococcus, and why it serves as an excellent model system to answer some of these questions. And second, I'm going to provide two examples of using Prochlorococcus as a microbial biosensor of both ocean biome properties and ecosystem transitions. So let's start out with our intro to Prochlorococcus. So not only are microbes ubiquitous and diverse, but they are also critical for ecosystem processes. In the marine environment, heterotrophic bacteria and archaea remineralize organic matter into inorganic nutrients. And these nutrients are then recycled into the marine food web. Additionally, through both photosynthesis and chemosynthesis, autotrophic microorganisms control the flow of all external energy into the marine food web, as well as the flux of carbon to the deep ocean. Both of these classes of microbes are highly abundant. In the ocean, as size goes down, abundance goes up. For example, if we look at a liter of water, so if you took a 2-liter soda bottle, you cut it in half and you threw it in the ocean, typically you would not catch any fish, but you would catch about 1 million nanoflagellates, about 100 million cyanobacteria, and about 1 billion archaea and bacteria. Another way to represent this is if we look at a microliter of water, so you can see that photo there of what a microliter looks like, and we did some fluorescence-based imaging, all of those green dots in that photo are bacteria. And so, as I mentioned, these are very, very abundant. And because of their high abundance, they have an outsized impact on global processes. So according to both NOAA and NASA estimates, 70% of the world is ocean and 95% of ocean life is plankton. Moreover, 50 to 80% of the world's oxygen has been produced by the ocean over geologic time. And finally, 25 to 30% of total anthropogenic CO2 emissions have been absorbed by the oceans. So as we are all probably very familiar with here, the oceans are super critical for many biogeochemical processes, including the carbon cycle. And a key organism in the global carbon cycle is Prochlorococcus. So Prochlorococcus is a marine cyanobacteria, and it serves as an excellent model organism because it is highly abundant. It grows at up to 10 to the 5th cells per mil, and it is responsible, for a large percentage of net primary production and carbon fixation in the ocean. Additionally, we have a number of cultured isolates of Prochlorococcus where we have fully sequenced their genomes. And we can look at these genomes to see their differing gene content and adaptations. So for instance, in this paper by Coleman and Chisholm in 2007, they sequenced 2 Prochlorococcus isolates, and they could see that one of the 2 isolates had additional genes related to iron uptake, suggesting differing adaptations between these 2 isolates. And so we can extend these patterns to a global scale. And that is because Prochlorococcus is globally distributed from about 40 north to 40 south. And within Prochlorococcus, we can identify unique populations that show differing distributions. So for instance, in this case, we have our green population, which is more abundant at higher latitudes and colder temperatures. We have our yellow population, which is more abundant at lower latitudes and warmer temperatures. Now, if we sequence marker genes from these populations, we can see that they are different phylogenetic groups. And these closely related groups or clades within Prochlorococcus are often referred to as ecotypes. And that's because they share the same environmental abundance distribution along gradients in temperature, light, and nutrients. So putting all of this information together, we can develop a phylogenetic tree for Prochlorococcus. So here I have a conceptual phylogenetic tree to kind of walk you through what this looks like. So if we take fully sequenced genomes of cultured isolates, it reveals that at the base of the tree, so the deepest or earliest division in population, was driven by adaptations to light. We have a high light clade and a low light clade. Moving along the phylogenetic tree, the next division we see is related to iron. And then finally, closer to the end of the tree, we have phylogenetic divisions driven by adaptations to low temperature and high temperature. These are the different groups that we refer to as ecotypes. So because of their high diversity, Prochlorococcus can be sort of thought of as an aggregate of populations, whereby subtle changes in the environment drive the diversification and structure of the Prochlorococcus phylogenetic tree, as well as a phylogenetic hierarchy of traits. And so the aim of my research and my talk today is to show that through the use of Prochlorococcus clades as a model, we can use our knowledge of microbial genome content and diversification to inform our understanding of marine ecosystem processes. So in my first example, let's examine how we can use Prochlorococcus as a biosensor of global ocean biome properties. And I want to point out that a lot of this work that I'm going to talk about next was done in collaboration with a grad student in the Martini Lab, Lucas Eustic. He's my co-first author on our publication from this work, and he did a lot of the analysis and writing of the paper. So many thanks to Lucas. Could not have done it without Lucas. He's been a great collaborator on all of this. Okay, so to begin with, I want to introduce the primary global ocean observation network supporting this work, which is the Global Ocean Ship-Based Hydrographic Investigation Program. GoShip coordinates a network of globally sustained decadal hydrographic sections, which cover the oceans from coast to coast and from full depth, so from the bottom to the surface. And this observing platform was designed to detect changes in the inventories of heat, freshwater, carbon, oxygen, nutrients, and transient tracers over long periods of time. And so our goal here was to determine whether we could in turn assess global ocean properties on this platform using the marine microbiome. And so, as I said, to address our broad research question of examining global ocean properties, I headed the pilot BioGoShip metagenome project. And this consisted of 8 research cruise transects across the global ocean, or about 256 days at sea total. So this involved packing up on our global class research ships and deploying our sampling methods such as Niskin rosettes or flow-through sampling to collect water samples onto filters, which we then froze and brought back to the lab. Once our samples were collected and sent to the lab, I developed a novel high-throughput protocol to sequence the metagenomes of all the BioGoShip samples. So once we performed DNA extraction, we performed metagenome sequencing. And so briefly, metagenomes are randomly fragmented DNA sequences. So this tells us both the species composition of the microbiome as well as the functional gene content. And here for this study, we examined 971 surface ocean metagenomes. So this represented about 3.65 terabytes of data. It also was very high spatial resolution. So this first BioGoShip data set, we had about 35 samples for every 5 degrees of latitude. By contrast, other international and global examinations of the ocean microbiome, such as biogeotraces in Tara oceans, typically had around 2 to 3.5 samples per every 5 degrees of latitude. So this allows us, this high spatial resolution allows us to answer novel questions about biome properties. And the specific biome property that we were interested in this case was nutrient limitation. So nutrient limitation exerts a fundamental control on oceanic primary production, and in turn, the productivity of food webs, as well as the flux of carbon to the deep ocean. So traditional Blackman and Liebig limitations hold that low nutrient concentrations limit the growth rate of individual cells and also sets an upper limit on the amount of biomass that can be formed. So nutrient limitation is very important to these global ocean processes. Another way to think about nutrient limitation is what's known as the leaky barrel analogy. So in this case, the ocean is the barrel and plankton biomass is the water in the barrel. And so the amount of plankton biomass that can be formed is limited by the nutrient in lowest supply. So whatever, we can think of the nutrients as the different boards of the barrel, and whichever board is lowest, that's what's going to limit plankton biomass. And so in many cases, we think the primary limiting nutrients in the ocean are nitrogen, phosphorus, and iron. And traditionally, these limitations have been assessed using nutrient uptake experiments. So we know there is high variability in biogeochemistry across the ocean. And this plot is a little complex, but I'll walk you through it. So the background is phosphate concentrations, which again illustrate this variability in biogeochemistry. Each dot represents a nutrient uptake experiment where inside the dot is the color of the primary limiting nutrient, and the color around the edge of the dot is the secondary limiting nutrient. And these nutrient limitation or nutrient uptake experiments tend to be highly labor intensive. They're a lot of work, and they're also affected by chemical detection limits. And so what we saw in a 2019 study from the Martini lab was that within Prochlorococcus, it appeared that Prochlorococcus's phosphorus gene content was related to phosphate concentrations. So you're going to see this term coverage a lot in the upcoming slides, and it basically just means abundance. And what we saw was that at low phosphorus concentrations, Prochlorococcus had more copies or a higher abundance of phosphorus genes versus when phosphate was in low concentrations, then they had, higher concentrations, then Prochlorococcus had lower gene coverage or lower abundance of their phosphorus genes. And so this suggested that cellular adaptation to phosphorus availability might be informative of phosphorus concentrations. And so we wanted to ask, can we use the principles of cellular adaptation to detect ecosystem-wide nutrient limitation? And so to answer that specific questions, we used 720 stations from 8 ghost ship cruise transects, as well as other cruises, as well as 228 samples from Tara oceans and biogeotraces. And so coming back to our metagenome process, right? So we performed our metagenome sequencing, and what we next did was we mapped those metagenomes to some reference Prochlorococcus genomes, and then we identified Prochlorococcus nutrient uptake genes. So we grouped the genes by different nutrient uptake types. We then had to do some normalization. So we took that nutrient gene coverage and normalized it by single copy core gene coverage, which is basically just an abundance-based normalization. We then transformed that into a z-score, which allowed us to sort of normalize across all of our cruises and samples. And then finally, we grouped those genes by different types. So we do group them by nutrient type as well as sort of a severity of nutrient stress or nutrient limitation in order to create our nutrient stress metric, which we call omega. And so we had 3 different types. We had nitrogen stress, phosphorus, and iron stress, and we were looking at genes that were indicative of both high and medium levels of stress. And so when we took all of these gene coverage values from across all of our samples, we then summarized them using a principal components analysis. So each of these little gray dots, which may be a little hard to see, but represent all of the stress genes within a specific sample. So each dot is a specific sample. And the vectors represent the trends across and between the different samples. So in this case, the vectors represent our different stress genes. And then where we have omega is where we have those composite metrics. And what we saw is this interesting tripartite partitioning of the ordination space, right? So all of our phosphorus stress vectors in this blue color are pointing in this direction. We have nitrogen stress pointing in this direction and iron stress pointing over here. And so we hypothesized that this suggests that there are trade-offs between the different types of nutrient stress. So in other words, populations are mainly nitrogen stressed at intermediate iron supply. They are mainly phosphorus stressed when you have high iron supply and high nitrogen fixation rates. And then populations are mainly iron stressed when you have macronutrients, phosphorus and nitrogen supplied by upwelling. And since each dot represents a sample, we can also use this tripartite partitioning of the ordination space to divide it into samples that are mainly phosphorus, nitrogen, or iron stress, as well as samples that show a certain level of co-stress between the different nutrients. We can then take these characterizations and back project them onto our global map. And what we can see is that the back projection of the nutrient stress characterization of our samples allows us to assess ecosystem state on a global scale. So what we observe is that there is a systematic variability in nutrient stress across the global ocean that's consistent with major oceanographic features. So for instance, most of the gyres are nitrogen stressed. In upwelling regions, such as in the equatorial Pacific, we see iron stress. In regions that have previously been, such as the North Atlantic, characterized by phosphorus stress, we can see that phosphorus stress is widespread. And what this does is highly extends the spatial resolution of our understanding of nutrient stress on a global scale. So here's that sort of complex plot from before where we had all of these very labor-intensive nutrient uptake experiments, and we can see that we can greatly extend our spatial resolution of our understanding of nutrient stress and nutrient limitation. We were also interested in verifying our indicator through a variety of techniques, and perhaps most convincingly, we saw a quantitative relationship between both the high and medium stress metrics and in situ nutrient concentrations. You can see that we have this negative relationship for both phosphate and nitrate, and our N stress and P stress metrics that was highly significant. This both validates our omega metrics, but also provides a basis for using genomic information as a quantitative indicator of biome properties and ecosystem state, especially when nutrients are below detection limits. So coming back to our question of can we use the principles of cellular adaptation to detect ecosystem-wide nutrient limitation and nutrient stress, we found that genomic content provides a systematic assessment of nutrient stress ecosystem properties at global scales. And so these principles are likely fundamental to adaptive processes in microbes and might be applicable across study systems. Our future directions for this work includes extending some of these principles to either other components of the microbiome or other components of the marine ecosystem. So specifically, our work so far has only focused on nutrient limitation in the photic zone, but rising temperature and deoxygenation may affect processes such as carbon export and other depth-dependent biogeochemical dynamics. And exploring genomic content at depth may inform us of both the current and future ocean state. So the future of the Pilot BioGoShip project is now funded by via NOAA, by NOAA, Alyse, and OER, as well as the NASA Ocean Biology and Biogeochemistry program. And this pilot project will be extended to examine marine microbiomes as well as other biotic measurements throughout the water column on GoShip cruises in the Pacific, Atlantic, and Indian Ocean. So, in addition to acting as an indicator of biome properties and cellular processes, microbial populations at high genomic resolution may also act as an indicator of transitions between different ecosystem states. And in particular, we were interested in using Prochlorococcus as an indicator of changes due to the 2014 through 2016 marine heat wave in the North Pacific Ocean. So between 2014 and 2016, the 2015 El Nino event, which you can see represented here, as well as the 2014 North Pacific warm anomaly, which was commonly called the blob, you might remember this, led to widespread and prolonged regional warming. This resulted in the warmest 3-year period on record in the California current ecosystem. Moreover, this prolonged marine heat wave resulted in somewhat unusual dynamics in the California current ecosystem. For example, in this paper by Kelly et al., they looked at export production. And what they saw was that although export production generally decreased during the marine heat wave of 2014, 2015, and 2016, export production north of Point Conception in California, so Point Conception is this sort of feature right here, was actually more similar to most non-El Nino years. So up at the top, 2006 was a non-El Nino year, and 1998 was a strong El Nino year. And so you can see this export production during the marine heat wave didn't exactly match the export production in 1998. Other studies have looked even further back, such as this one by Thompson et al., which looked at trends in larval fish populations and their responses to El Nino between 1950 and the 2014 through 2016 marine heat wave. And what they found was that anchovy and sardine populations actually showed the opposite pattern compared to the past 60 years of El Nino trends. So anchovies, which they expected to decrease, actually increased during this marine heat wave, and sardines, which they expected to increase, actually stayed relatively low. So there are some unusual dynamics going on here, and so given both the prolonged warming and the unusual impacts of the 2014 through 16 marine heat wave, we wanted to know whether we could use cyanobacterial communities as a biosensor of this marine heat wave in the Southern California Bight. So to answer this question, we decided to examine Prochlorococcus and Synechococcus trends using time series data from the Newport Beach Pier, which is highlighted here in red, in Orange County, California. And so to understand our hypotheses here, I have to introduce you to Synechococcus. So we now know a lot about Prochlorococcus, and Synechococcus is Prochlorococcus's sister taxa. So whereas Prochlorococcus has smaller cell size and is more abundant in high temperature and low nutrient conditions, Synechococcus has slightly larger cell size and is in higher abundance in colder temperatures and higher nutrient conditions. Nevertheless, they have very similar phylogenetic structure within each of these genera. So they both have some low temperature clades and they both have some high temperature clades. So given these dynamics, we predicted that in response to the marine heat wave, we'd see an increase in Prochlorococcus relative to Synechococcus. We'd see an increase in the relative abundance of these high temperature ecotypes, and we'd see changes in sub-ecotype populations. So I'm going to be using the term haplotypes to describe these populations. And a haplotype is a single nucleotide polymorphism delineated population or clade. So we're using changes at the nucleotide level to delineate these haplotypes. And I'm going to talk about that a little bit more in a minute. So our methods for determining whether we saw shifts in cyanobacterial populations over time at the Newport Beach Pier were, first, we collected temperature, nutrient, and microbial genomic DNA samples from the Newport Beach Pier between September 2009 and December 2018. Collections are actually ongoing, but we're just going to focus on this approximately 9-year period. This represented 434 time points, so approximately weekly, a little bit less than weekly. And we performed RPOC1 gene sequencing. So that's a specific amplicon. So in contrast to our metagenomes, in this case, we take our microbiome sample, we extract the DNA, and here we're just looking at a single gene. And this is our RPOC1 gene is going to inform us of the relative abundance of our cyanobacterial populations. So at the Newport Beach Pier, we were able to identify significant seasonal and interannual trends in both temperature and nutrients at our study site. So here we have the overall time series of temperature and nitrate. And we could characterize this using an ANOVA-based analysis. And so that's what we see here on the bottom. So these are our monthly trends and on the left, and our yearly trends on the right. And so what this represents is any value that is above 0 means we see a trend that is above the overall average or an increasing trend. And if we see values below 0, that means that we see a trend that is below the overall average or a decreasing trend. And so to start out, we can see very typical seasonal dynamics, right? So in the winter, we have lower temperatures and higher nutrients. And in the summer, we have lower nutrients and higher temperatures. So we can then extend that to our interannual trend. And what we see is some very interesting dynamics. First off, nitrogen at our study site actually decreased prior to the 2014 through 2016 heat wave. And we think that might be due to regulations of nitrogen runoff in Southern California. But of course, we're not entirely sure. And then we do see our increasing temperatures between 2014 and 2015 and beginning to decrease as we move into 2016. So this is pretty much what we expected to see in terms of our abiotic or environmental trends. And we also saw during the warm period a significant shift to Prochlorococcus relative to Synechococcus. So here we have our Prochlorococcus clades in green and our Synechococcus clades in purple. And what we can see is right around 2014, this significant increase in Prochlorococcus relative to our Synechococcus populations. We can also apply the same ANOVA-based analysis to see whether these clades are increasing or decreasing across the time series. And what we saw was that all Prochlorococcus clades were increasing, as well as Synechococcus clade 2, which is one of the Synechococcus high temperature clades. So indeed, we observe an increase in the high temperature ecotypes across this time series. So we also wanted to look into these higher temperature ecotypes in more details to see if we saw subpopulation shifts. And these plots are very complex, so I'm going to take my time in walking you through them. So each branch in these clusters actually represents a sample. And for that sample, we have the consensus sequence for the Prochlorococcus highlight 1 population or the Synechococcus clade 2 population. And we can identify single nucleotide polymorphisms in these populations by comparing them to a reference sequence. So for Prochlorococcus highlight 1, we had this MIT 9515 sequence, and for Synechococcus, we had this reference CC9605 sequence. And so by comparing these consensus sequences to the reference, we can identify single nucleotide polymorphisms, and then we can cluster those single nucleotide polymorphisms. And in both cases, what this suggested was that each of these ecotypes had 2 distinct sub-ecotype or haplotype level populations. And what we could then do is look at the relative abundance of these haplotype populations across the time series. And what we saw similarly was a big shift to between these 2 different sub-haplotype or sub-ecotype populations within Synechococcus and within Prochlorococcus that simultaneously occurred starting around 2014. So again, in Prochlorococcus, we see a shift from haplotype 1 to haplotype 2. And then within Synechococcus, we see a shift between haplotype 1 and haplotype 2 within Synechococcus. Now, within Synechococcus, we saw a brief return to haplotype 1, approximately during 2017, but then it also went back to haplotype 2. So by the end of this study period, we had not seen a full return to pre-El Nino or pre-heat wave conditions. And so we were also interested in what could be possibly driving these dynamics, or where these populations could potentially be coming from. And so to see where these populations might be coming from, we compared our RPOC1 sequence from the Newport time series to a previous cruise that occurred in the North Pacific Ocean, going from Hawaii to the equatorial regions. And what we saw was that Synechococcus clade 2 haplotype 2, which if you remember, we saw sort of in the latter half of our time series, was closely related to samples taken from the North Pacific subtropical gyre. In contrast, that Synechococcus 2 haplotype 1, which we saw in the earlier part of our time series, was more related or more closely related to samples that were taken in the equatorial Pacific. Now, the equatorial Pacific also has equatorial upwelling and has higher nutrient conditions. So this suggests that these populations that we're seeing at Newport are related to the broader populations of Synechococcus across the entire Northeast Pacific region. And so we hypothesized that the Southern California bite is potentially a transition zone between microbial populations. So the California current ecosystem is actually influenced by 2 main currents in this region. There's the California current, which has colder, lower salinity, younger, lower nutrients, and more temperate waters, versus the California undercurrent, which is generally warmer, higher salinity, older, actually higher nutrients, equatorial and subtropical waters. And these two currents shift over time in their influence on the region. So Bograd et al. Used this silicate-based tracer or silicon-based tracer to detect changes in the California undercurrent signal. And what they saw, that paper saw, was that between 2008 and 2012, there was actually an increase in the California undercurrent signal. So in the future, we would like to do more work to better link these microbial dynamics to these tracers to better understand how changes in the Southern California microbiome is influenced by ocean physics. So coming back to our research question of can cyanobacteria communities be used as a biosensor of the impacts of this marine heat wave, we saw both Prochlorococcus and the high temperature ecotypes from Pro and Syn both increased in abundance. We also saw that cyanobacteria composition from the genus to the haplotype level classification did not fully return to pre-El Nino status by the end of 2018. And a combination of, we hypothesized that a combination of both in situ warming and changing water masses was likely driving a cyanobacterial composition shift in this region. So our future work, we received a grant from the Joint Genome Institute, which is funded by DOE, for metagenomic sequencing across this time series, which has already been completed. So we have about 250 short read metagenomes, so that's about bi-weekly resolution of metagenomes, as well as 4 seasonal long read metagenomes from pre-El Nino and heat wave conditions, and 4 seasonal long read metagenomes from post El Nino and heat wave conditions, as well as 192 single cell metagenomes to help us assess both the rest of the microbial community, as well as determine how shifts in functional gene content responded to this 2014 through 2016 marine heat wave. So in conclusion, I would say that microbial genomes are a powerful indicator of both spatial and temporal changes in marine ecosystem processes. And recent improvements in both sequencing and computational techniques have allowed us to derive quantitative relationships between microbial genomes and the environment. I'd say we are truly standing at the entrance of a new omics era in marine microbiology and biological oceanography that I hope are going to allow us to better assess and understand how our ocean is changing. So with that, I'd like to thank you for your time, and I will take any questions.
Alyse, thank you so much for your great presentation. Very informative. And I hope we all learned lots. We don't have any questions at this time, so please feel free to post them in the chat or in the questions for staff box or tab on the GoToWebinar screen. I do have one question though to get us started. So when looking at Prochlorococcus as a biosensor, you found differences in nutrient stresses based off of the gyres or upwelling or physical attributes of the ocean. And I'm wondering if any of those attributes have been, and the data you've used have been combined with satellite data of these different aspects of the ocean, and if that could ultimately be used for forecast modeling in changing of these microbiomes, like you kind of covered in the second half of your presentation. So ultimately, do you see this data kind of being combined with satellite data for forecast modeling, particularly of these types of changes for El Nino events?
Yes, that's an excellent question. And actually, that's a really important future direction for the pilot BioGoShip project. So one of the partners in funding the future of BioGoship was the NASA OBB program. And so on these, so there's been 2 cruises that have already occurred, PO2 in the Pacific and A16 North, which just concluded in the Atlantic. And then there's one coming up this summer, IO5 in the Indian Ocean. And so this data set was only surface ocean samples and was largely limited to metagenomic sequencing. We didn't have a ton of other biotic measurements on these early cruises. But in the upcoming cruises, in addition to the omics, they're also taking a number of cellular health and pigment and fluorescence-based measurements concurrently with the metagenomic samples. And the hope is to link this genomic nutrient stress metric as well as sort of future stress metrics to these pigment-based analyses of cellular health, and then in turn link that pigment-based analyses of cellular health to satellite-based measurements to do exactly what you're talking about, sort of both assess sort of different metrics of cellular health as well as nutrient stress and carbon dynamics, link all of that to satellite-based metrics and thus extend the reach of our projections both now and in the future. So yes, is the short answer, is that is a really important future direction of this research that is underway.
Yeah, I'm already looking forward to hearing some of those outcomes. So we do have a few questions that came into the chat, and we'll go ahead and start. The first is, in your first example of the green and yellow strains of Prochlorococcus, what do you think accounts for the abrupt rather than gradual change in distribution?
Yeah, that's a good question. So I think it relates to their thermal tolerance, right? So these different populations have different thermal niches and temperature growth optima. And what we see is that at 19 degrees Celsius is sort of the crossover point in growth rates between highlight 1 and highlight 2. Those are the 2 different high temperature clades, highlight clades with different temperature adaptations. And so that really abrupt change tends to occur around 19 degrees Celsius, where we see this crossover between highlight 1 growing faster than highlight 2. Sometimes there in the environment there is more of a gradient because we see a bit of a lag temporally to responding to changes in temperature over the seasonal cycle, but I think it's largely related to these different growth optima of the 2 different clades.
Thanks, Alyse. And that question was from Eric. From Kelly, our second question, is there a plan to further apply or test the omega indicator?
Yeah, well, so we are planning to do that in different ways. So there's the plan to link the omega indicator to satellite metrics, which I mentioned before, but also we want to look, so our measure, our patterns are currently, that I talked about in this talk, are spatial patterns, right? So single time points taken across the global ocean, but we also want to look at this omega indicator in our Newport time series to see if it shifts over time in a single location. So we want to better understand temporal dynamics of nutrient limitation in addition to spatial dynamics of nutrient limitation or nutrient stress, really. So yes, there's plans to, like I said, both link it to satellites and look at it over time.
Thanks, Alyse, and thank you, Kelly, for the question. This question is from Luke. There's two parts. So the first is, you mentioned that the gyres tend to be iron limited, but also that the North Atlantic is phosphate limited. Do you think that is because the North Atlantic gyres receive Saharan dust, whereas other gyres do not?
So a little bit of a clarification. The gyres tend to be nitrogen limited. Here, let me go back to the slide. Let's see if I can do this. Let me do it quickly. Let's see. These are all slides. Let's see if I can go back. Okay, so the gyres tend to be actually nitrogen limited, and so I'm trying to remember all of the components. So of that question, but what we think is happening is, I'm actually going to show you this PCA again, is that Prochlorococcus use nutrients at a stoichiometric ratio above the vertically supplied nitrogen to phosphorus ratio. And this leads to a default state of residual phosphate for Prochlorococcus and corresponding nitrogen stress. And so this means that populations are mainly nitrogen stress, as I said, when there is at least some intermediate iron supply, which could be largely dust deposition in certain regions. But then what we see is that when we have nitrogen and phosphorus supplied by upwelling, that's when we have a state of iron stress. And then when we have nitrogen supplied by nitrogen fixation, that's where we see a state of phosphorus stress. So basically, you're nitrogen stressed when you have iron, you're iron stressed when you have phosphorus and nitrogen, and you're phosphorus stressed when you have some iron and nitrogen supplied. So it's this, three-way dynamic between supply and corresponding stress. I know that's kind of a complex answer. I don't know if I addressed all components of the question there, but yeah, so it's some complex dynamics related to, supply and demand.
And the question had proposed possibly the North Atlantic gyre receives Saharan dust, and that's maybe why it's not phosphorus limited.
Yeah, that could definitely be a contributing factor, right? So because they're receiving iron and we have nitrogen fixation in the region, that's why we're seeing phosphorus stress in the North Atlantic.
And then we have one last question for today, also from Luke. Do you think that metagenomics will be able to capture the fine-scale haplotype trends that you discovered in the gene-based metabarcoding in Southern California?
Yeah, so I guess there's two fine-scale trends. So that's a good question. We did develop, so Lucas, who I mentioned earlier, did develop a pipeline to extract RPOC1 from our BioGoship metagenomes and align it in order to look at sub-ecotype populations, and then also link that to functional gene content. It's definitely a bit of a challenging pipeline, but it's looking promising. And so we were able to see that the metagenome-based sub-ecotype populations had a distinct phosphorus stress, like a clade that was associated with the high phosphorus stress regions, where we also see an increase in our phosphorus omega metric. And so there is some indication that, at least at a certain level, I don't know, what I don't know is if it's those populations are that we extract from the metagenomes are at as high of a resolution as those that we extract from amplicons, but we do see some correspondence between our nutrient stress metrics and some sub-ecotype populations for our GO-SHIP metagenomes. So I do think there's some potential there, but of course there are limitations in sort of the scale of phylogenetic resolution that you can get from metagenomes due to, right, uncertainty in the mapping algorithms. So I think there's potential, but it needs to be those types of investigations need to be interpreted with caution, is what I'd say.
Awesome. Thank you, Alyse, so much for your time today and your great presentation. Thank you all for joining. We look forward to seeing you at our next seminar, which occurs on the third Wednesday each month. Thank you all so much, and we will see you next time. Thank you, Alyse.
Thanks, Nicole.